welcome.
Hello, everyone. If you'll bear with me, and thank you, Cam and Graeme, for being dead on time. I should have mentioned that I usually allow just a couple of minutes for the online crowd to join us. They often don't all hit us in one hit. So with your forbearance, I'd just like to hold proceedings for a minute and then we can proceed from there.
No worries.
While you do that, Dave, I might just acknowledge Miranda's work as well for putting the seminar on today. She was the driver behind having this hybrid session. So we're here in person today because of Miranda's hard work. So thank you, Miranda. Appreciate it. Just to acknowledge your work on that as well. Thank you.
Okay, we might kick off our proceedings then. Welcome everyone to you all to our monthly online research seminar. It's good to have you with us again. And for those of you that joined a couple of minutes ago, Dr. Cameron Welf, our local MC, mentioned that this is a hybrid seminar. So we're mixing our online audience, which we expect will number up to about 100, with our physical audience located at QUT in Brisbane as well. So a warm welcome to both our online and our face-to-face audience. Just want to echo Cam's comments. So first of all, let me thank Associate Professor Miranda Mortlock and of course, Dr. Cameron Ralph, who are there on location for their efforts in coordinating the local event. And to Chris, just met you, but thank you. You're our local IT mastermind and it's good to have the online and the virtual meshing so nicely. For those of you I haven't met before, my name is Dave Lamb. I'm the Chief Scientist of the Food Agility Cooperative Research Centre, and I'll be your hybrid MC until our online component finishes on the hour. And then, of course, Miranda and Cam will continue with any interactions with the audience on location. So just to echo Cam, I would like to take the opportunity to acknowledge the traditional owners of the many lands and waters on which we both face-to-face and virtual audiences live and work. Of course, the traditional First Nations owners of the lands where QUT sit, of course, are the Turrbal and Yagara nations. I'd like to particularly acknowledge those and pay respects to elders of all the lands and waters, both past and present and emerging. So those of you who are familiar with Food Agility, we are a cooperative research centre dedicated to unlocking the power of digital to transform our agri-food sector. Now, before I introduce our host, our speaker, just to remind you of some brief housekeeping. First of all, we are recording this session and we will make the recording available to you via an email link after the session has concluded. We do welcome questions. Please use the Q&A tab, not the chat tab, but the Q&A tab located at the bottom of your screen for those virtual audience. And of course, for the face-to-face audience, unless you'd like to have your question raised and aired with the virtual audience, which can be done through Cam or through yourselves going to the online Q&A tab, then there'll be an additional question and answer session once the online component ends. So a reminder to you all, please use the Q&A tab. And then at the end of the presentation, I'll be having a conversation with the presenter and we will exchange those questions and continue the discussion. Climatic variability in dryland production environments generates crop production risks. Every year is different and even less predictable, thanks to, among other things, anthropogenic global warming. Optimal combinations of genotype and management vary amongst sites and seasons, and experimentally assessing the spectrum of potential genetic by management by environment combinations which constitute the adaptation landscape in which we're growing our food is prohibitively expensive both in dollars and resources. So robust crop models provide an avenue to predict likely outcomes and production risks via simulation with a whole range of climatic scenarios. And this is the topic of today's seminar. It's with great pleasure that I introduce to you our speaker, Professor Graeme Hammer of the Centre for Crop Science, Queensland Alliance in Agri-Food or Agriculture and Food Innovation, otherwise known as QUT, at the University of Queensland. It's a privilege to have you here, Graeme. Graeme conducts research on the physiology and genetics of complex adaptive traits in field crops, with a focus on water productivity in cereals. His research underpins the development of mathematical models of crop growth, development and yield that enable simulation of the consequences of genetic and management manipulation of crops in specific target environments. Graeme has and continues to play a leading role in the design and ongoing development of the Apsim crop modelling platform, which is now used worldwide, and I'm expecting that he's not a stranger to many of us in the audience. Graeme's a fellow of the Australian Agricultural Institute, and in 2012 was awarded the Farrer Memorial Medal, followed by, in 2013, the Australian Medal for Agricultural Science. So again, it's a privilege to have you with us, Graeme, and we look forward to hearing from you. Over to you.
Thanks very much, David. We'll just get this system working. Okay. Thank you. Thanks very much. And thanks very much for the invitation, and thanks to Miranda for organising. I'm quite happy to be a hybrid and have an audience in the room and online. And I think David's given you a pretty good background of the sort of work I do and the topic that I want to talk about on crop design for adaptation. And I just did want to reinforce that my work is very much about field crops, and particularly dryland agriculture and broadacre field cropping. And that's sort of the focus, although the Apsim activity in modelling goes much broader than that. But I want to talk about crop modelling and simulation in the context of designing crops for adaptation to these variable environments. But I want to start with a take-home message that comes from a piece of art, if I can get this working, which I liked, and it's actually a sculpture in the sculpture park in Des Moines, Iowa. I was on a visiting scientist sabbatic at Pioneer Hybrid, who make actual hybrids, and are one of the biggest seed companies in the world. And this sort of struck me as a nice parody of Rodin's Thinker, which many of you may have seen in Paris. But I thought this is interesting that this artist put a hare in here, and as a parody of that. But it also struck me as interesting in the area of work I was in, because I don't know if you've ever startled a hare in a field, but if you do startle a hare in a field, it just darts off, okay? And it sort of just darts off anywhere. And so it's like it's unthinking. So the take-home message is get the hare to think about the journey before running off. And I'll come back to that. So there's a bit of a contradiction about a hare and thinking, which struck me when I saw this piece of art. And so the main point about that is, how do we actually integrate advances in digital and predictive capacity with biological technology to transform crop design? So let's think about how to do it before we just duck off and start doing it. And I want to come back to the point about transdisciplinary and transcale interaction, about, in sort of the context of how to do that well. So given that's the take-home message, I've got 6 parts I want to talk about. I want to start by just giving a little bit of background about what an adaptation landscape and genetics by management by environment interactions in crop production actually means, briefly. I'll cover a bit about the problem of prediction and production risk and where crop simulation might fit into that. And I know many of you are involved in prediction problems, so this is a specific instance. I'll cover a bit about the Apsim modelling platform, seeing as I've spent my life actually helping to build it, and go inside some detail on how we go about modelling crop growth and development, and the research and data requirements that underpin doing that properly. And then give an example of an in silico study for crop design for adaptation, where I'll use sorghum, which is a crop I work on a fair bit. And then just come back to some broader implications rather than my specific example. So if we think about the adaptation landscape, I like to view that as this sort of picture where you might think of the complexity of maybe the axes on here might be genetic traits and management alternatives, and the vertical distance is the fitness. And so in ecology, this might be called a fitness landscape. In crop improvement, we'd call it an adaptation landscape. Whereas what you really want to do is predict how to get to these peaks. So presumably the peaks are where you want to be. They're the highest performing, most fit positions. But the problem is this isn't a stable landscape. It's more like an ocean. So this might be the outcome in one season. If you change the season, these peaks just wobble around, okay? So what's a peak in one season may be a valley in another season and vice versa. So you've got this whole landscape confounded by environmental variability. So how the hell do you go about predicting where you are and navigating to where you might want to be? And that's a general problem of crop improvement. So prediction is a pretty key part of all this, because you want to be able to predict the outcome, and let's say phenotype, or we might just say, let's say yield of a crop. You want to be able to predict what might happen given various combinations of genetics and management activities. And there's a sort of black box in the middle that says, well, if we know a whole lot of stuff about this, then we should be able to do something that will predict that. And there's been a whole lot of developments in statistical methods, for example, that are used conventionally to do this, in terms of predicting this phenotype if you're a plant breeder from genetics and environment and G by E interaction, or if you're an agronomist, maybe from the management and environment and management by environment interaction. And so this is sort of classical statistical analysis that has worked and has generated improvements historically over decades, but it's slow, okay? And it's very empirical, and it relies on data, comprehensive data of those things. And so you can only predict really what's inside your data sets in an empirical way. So can we do better than that? And what are the advanced approaches to prediction that we might employ? And so one of the things that strikes me, and this comes back to the hair, is there's a whole lot of stuff now about being drowned in data. And so we get genetic data, phenotypic data by the bucket load, or nowadays, I suppose, by the terabyte. And this is another piece of art. This was it's actually a waterfall of genomic data. It was actually running down the wall. This was at Mona in Hobart. And my wife snapped me standing in front of it. And I thought, it's a pretty good sort of example, again, of just drowning in data. And I do a fair bit of work with geneticists, and it's a bit like that. And so, you know, you can sequence lots of things, you can image lots of things, you can run UAVs over the tops of crops, and you can get all sorts of hyperspectral reflectance data, and you've just got data. And so, fantastic. And people sort of get lost in the technology of getting the data and don't think too much about what to do with it. And so suddenly we've now got big data, and we need all sorts of big data solutions. And so there's a whole lot of work on novel algorithms to deal with this in a predictive sense. And that's fine as far as it goes. But as far as I can see, we haven't really got to the point of capturing biological insight in the way we go about doing this. And that's what I like to talk about. Can we introduce biological insight into this sort of big data, or the other way around, bring some of the big data capacities into the sort of biological insight methods to do this whole prediction system better? So when we come back to the black box, what we can do is put a crop model in here, okay? And that's what we've been playing with. And this is just a sort of schematic of a crop model, and all the interactions of the nodes in here among the various components of a crop, or the attributes of the system. You capture the dynamics of the system through algorithms that describe, quantify the linkages. And so you've got environmental effects and crop attributes that you generate over time. So the idea here is to put some sort of dynamic crop model in the centre to help this G by M to P prediction. So the third thing I then want to talk about is, well, what sort of models might we use? And I said, we've been playing around with the Apsim platform for an eternity. And I'll just briefly give you a little bit of background on that, but there's a nice paper that describes it in some detail. And this had a long history of development in terms of this prediction problem. And it's sort of evolved with the nature of that prediction problem. And so I'll give you a bit of a timeline just to show you it's either you think it's really old or it's actually taken a long time to emerge and develop. But we started this group back in the early 90s, and it was a research group between CSIRO and then the Queensland Government Department of Primary Industries, who I was working with. And we kicked this thing off and started building a framework for agricultural production system simulation. We were mainly interested in management actions, not so much genetics. This went through a few incarnations of activity and cooperation amongst organisations. UQ became involved when I moved there. We had a team of good software people and good software engineering practice to combine the science. This with good software. This merged into, rather than a formal research unit, more of an agreement among the partners to continue the development of this software platform over a period of time to present, and a number of other partners have joined. And so we have this sort of science software development of this system simulation tool. And there are now quite a few members, and it's really an international activity to maintain and develop this cropping system simulation platform. There are a number of organisational committees to run it. So there's a steering committee which actually sort of sets the policy framework around what it does, and there's reference committee which really looks at maintaining the quality of the science and the software and the code in the platform. And so this is a managed system. There's good version control and all the rest of it. And it's now used, it's got widespread use globally for all sorts of things. So the philosophy with this model, though, wasn't so much about let's just predict yield of a crop. It was about let's look at a cropping system over time. And so there's a very big focus on the soil. And so the soil's the one thing that doesn't come and go. The crops come and go, the management changes, the environment changes. The soil will change its properties, and you can affect those properties by how you manage and what you do. But the soil's always there, so it's a central focus in our modelling system. And then you have all these components that you can bring in from the toolbox to look at various components that affect how a crop or the soil works. And so all the science sits in all these pieces. You put the pieces together, you can simulate a field or you can simulate a whole farm. And so that's a sort of way it sits together. There are a large number of modules for various field crops, and these get added to. It's very much, there's not a structured development in the sense that we say, oh, we want to now go and do a model for this crop. It depends on funding. And so these are sort of, the system is just an organisation around an amorphous set of projects that uses this underpinning sort of knowledge bank system and adds to it as you go. So when one gets a project to work on maize, you think, okay, I'll build a maize model and put it in here, and that will help me do that project. You usually, at the beginning of this, we didn't tell the funding body that because they didn't want to talk about models, but they liked the outcomes. And so you could use a bit of stealth and get this thing happening. And the same with all the soil and animal climate routines. So I want to talk a bit about, go inside a little bit, the modelling of the crop part of this. And so you can sort of see inside a little bit just what's needed. And if we think about a crop model, this schematic I like just shows all the processes going on in the soil, plant, atmosphere system in relation to a crop capturing resources, be they light, water or nitrogen, how efficiently it might use them and how it partitions them to whatever product you want. And so there are a whole lot of component processes that you need to quantify to be able to get this sort of yield as genotype by environment, by management interaction predicted. So ultimately what we're after here is that prediction. And so this is a very messy diagram of how this works. And so it's a sort of spaghetti diagram, but you don't really need to understand the detail, but there'll be a quiz as you leave. And so the idea here is this is really just the sort of interactions of the components of a model and how it hangs together. And I'll just lead you through a few bits of it because it's not that scary. And so these are all the interactions and feedbacks based around this concept of physiological determinants of crop growth and development. And the top line up here are just all the environmental inputs: radiation, temperature, vapor pressure, rainfall, and so on. Soils over here, the crops over here. And phenology is a key component in here. You have to be able to predict the progression through the life cycle of a plant. And to do that, temperature and photoperiod driven will enable you to see how much leaf area that plant or crop is going to produce, affects total leaf number, affects leaf area. So you predict the progression through the life cycle, you predict the development of the leaves and the canopy and branching, and that gives you the size of the canopy. Once you know the size of the canopy, you can actually say, well, how much are you going to grow? And how much light are you going to intercept? What efficiency are you going to convert that light into carbohydrate? And that connects back to water. If you've got plenty of water, that's fine. If you don't have enough water, then water can limit how much growth you can get. Once you get beyond determining how much growth you've got and accumulated, and you've got to flowering, you then say, well, okay, how much of that is going to be allocated to grain and how rapidly are those grain going to grow so I can predict the grain yield. So each of these arrows are a set of equations that determine the dynamics of how these things interact. And so you get the interactions with the environment, the interactions with the attributes of the crop. So these things in the grey here are actually like genetic attributes of a crop, which genetically one can modify. And management will change a number of these things as well. So you can get genetics, management, environment, and all the interactions and make predictions. Just takes a while to build that. And you have to do a little bit of work in terms of how things respond to different factors. And so here's a sort of classical piece of work on temperature responses for development rates. It comes out of the group in France, and they put a whole lot of sort of meta-analysis together. And these are sort of basic temperature response for a whole lot of processes for a whole lot of crops. And so, you know, you just have to know that. You have to be able, okay, this is an empirical fit to that, but you can actually put coefficients on that have some meaning, and you've got a standard temperature response of what processes are going on. So if you know the temperature, you can work out the rate that these things are going to be happening. It's also affected by photoperiod. So in addition to this thermal time, temperature-based notion, you've got photoperiod responses. So this is a short day plant where it reaches reproductive stages quicker under shorter photoperiods than it does under long photoperiods. And you've got experiments that just show that, and you can quantify it. So you can capture the biology into some simple equations at each of this process level. To get from there to crop growth, the increment in biomass, you have to think about this capture of resources. And I'm going to ignore nitrogen, although it complicates things, but it can be dealt with. And just think about water and light. And if we've got how much crop growth is going to happen today, it's either light limited or water limited. If it's light limited, how much light am I intercepting? how efficiently am I using it? And this is where I've got plenty of water. If I don't have enough water, how much water am I getting and how efficiently do I convert that? And so it's a fairly simple sort of way of specifying this, but it sits back into some really good theory on stomatal regulation and the trade-off between capture of CO2 and loss of water. And the equilibrium internal CO2 concentration in a leaf determines these efficiencies. So even though that looks like a very simple construct, thanks to Graham Farquhar, we've got this beautiful theory that connects directly to that. And so you can then measure those things, so you can quantify them. So you can do experiments and look at how much light is intercepted, and there are attributes of the canopy. So this is the area of leaves on a crop canopy and the fraction of intercepted radiation, which you can measure, and you can fit these negative exponential sort of equations, and you get this extinction coefficient, which in fact in physics is just really the leaf angle distribution of that canopy and how much light it intercepts given a certain area of leaf. So you can see even when you've got a leaf area of 2, that's 2 meters squared of leaf per meter squared of ground. It's not, you're only intercepting 60% of the light, okay? Because your leaves are doing a lot of self-shading. They're not flat bits of paper. So once you then know how much light is being intercepted, and that extinction coefficient can change. If you've got a crop that does that or does that, you get a very different extinction coefficient. And you can then look at how much radiation is that crop intercepting over time. You accumulate it. You can look at the amount of biomass of that crop, and you'll get a very linear response. And this is the efficiency of conversion of that intercepted light to biomass. So these are sort of critical parameters that you have to go and experimentally determine to be able to put inside this sort of model. Given that, I won't go into the bottom bit, but you can, once you know how much you're going to grow, you can work out how much water you need to do it. But if you're water limited, it really depends on how much you're able to get out of the soil. And so and the efficiency with which you can convert that transpiration of water into biomass. And so that, again, there's this sort of intrinsic efficiency which comes back from that theory. And there are specific numbers for various species, whether it's C3 or C4 photosynthetic pathway. And we need to then think, how do we work out how much water is going to be extracted from the soil? And there's, again, some nice theory, some old stuff from Paziura, where if theta is your soil water content, time and days, this is the upper limit, sort of drained upper limit for a profile. This is the lower limit, as much as a root system can extract. And if you just watch that over time, it does this beautiful exponential decay. And so you can fit, again, an exponential decay equation to that, and you've got this decay constant, which happens to depend on the hydraulic conductivity of the soil and the root length density. And if you don't know either of those things, which you usually don't, you can estimate it by measuring what happens to soil water over time. And then you can accumulate how much you get from each layer, and that's the amount of soil water that the crop can get on a day. So when it's not able to get as much as it wants, that's as much as it can get, then that will dictate how much it can grow. And so you can put that in a model. And here's the sort of experiments you have to do to be able to work that out. And so these are measures of soil water content in different layers of a soil profile over time. And so this is at 50 centimetres, 90, 110, and so on, down to depth. So as you get down to depth, you'll see it doesn't start till later because the roots they take a while to get there. And it's not as effective because your root length density is not as great. And so, but once you've got this, you've actually quantified the characteristic of that combination of crop and soil to be able to extract water. Beautiful stuff for PhD students, which Mike Robertson was in those days. And so once you've done all that, you can sort of pull it all back together and say, well, okay, can we now predict? We've got all these bits and pieces and algorithms and equations, and we've got a sort of a schema of how they all connect based on sort of theory of crop growth and development physiology. Then you can say, okay, well, let's look at it. And these are experiments that were done with maize, looking at nitrogen treatments actually. But again, you know, sort of very detailed experiments where you harvest the plant every 10 days or so, and you can measure how much biomass it's got, how much mass there is in each of the components, how much nitrogen there is in each of the components, how much leaf area you've got, and so on. And then you can say, well, okay, given that we've now parameterized all those equations, what can we predict? And the simulation is the solid and the lines on here. And so you can see mostly it does remarkably well at predicting the dynamics and the amount and allocation of carbohydrate and nitrogen to the various organs. Crashes out occasionally, doesn't get the effect of nitrogen stress on leaf area quite right here, but generally that's a pretty bloody good job. And so that's fine where you've got high quality data for a small number of experiments. But if you want to simulate an adaptation landscape, you need to think, well, how is that going to extrapolate? Have I really just got it nailed for that experiment? But when I go over there, the answer is going to be ridiculously different and I've missed something. So the idea is to have high quality data across a whole range of conditions, and at least then enable you to do predictions and do these predicted observed sort of plots. You may not have as intense data as the previous slide, but you've got pretty good data. And you can predict each of these variables, time to maturity, the phenology, leaf area index, biomass, yield, reasonably well. And that's a pretty good outcome from a crop model. That's about as good as it gets. So I wanted to show this example with soil water prediction because I like it. I don't think it's going to work. No, it's not. There was a video there, but anyway, I probably won't have enough time anyway. But you can go into detail, and this thing actually predicts the soil water measurement over time of a dry down profile, and it does a very good job of it, which sort of suggests that that sort of supply-demand concept for predicting water use is pretty good. So given that we're able to build a model and we've got something that captures all those interactions and feedbacks, and it's got a good link to understanding how the system works and experimentation that's actually measured it. You know, what can we do with it? Now that we've got something that captures all these non-linearities and interactions, let's look at doing some adaptation analysis with it. And so I'll give you a couple of examples. The first one's sort of just a teaser really to just sort of say, well, let's look at, this is a sorghum crop, and I want to just look at a simulation. So this is a 100-year simulation. So each dot on here is an outcome for 1 year. So let's say there's 100 dots for each strategy. The standard strategy is what would normally be done. A cultivar of a certain maturity, grown at a certain density, and with every row planted. So this picture over here is a skip row sorghum system where there've been rows left out. And so it's a more conservative water using system. Conventional system is those rows are not skipped out, and that's the solid planting system. So, and that's the standard along here. Then we look at a high input system and a low input system and plot the yield of those against the yield of the standard. So the only variable in here is the seasonable weather. All the starting conditions are set at the same. And if you look at the high input system, what you've done is extend the maturity, increase the density. So basically it's a bigger leaf area, bigger canopy system, trying to go for higher yield. And under high yield conditions, it gets higher yield. But under poor yielding conditions, it gets poorer yield. So that's the blue line. The low input strategy is a much more conservative system. So what you do here is reduce your density, go to this skip row system. And what happens is you can get a much lower yield in the good years, a much better yield in the poor years, and so you minimise your risk. So if you want something every year, and you know you owe the bank a lot, this isn't a bad way to go. But if you're a bit of a risk taker, you might think, well, I'd want to get this one up here, but you don't know which year you're going to be in. So to extend that, what we've sort of looked at is doing that across the whole sorghum production region in Australia. And so this is the map of where sorghum is grown from northern New South Wales to central Queensland. And climatic variability, particularly rainfall variability, is critical. And the effective use of water is really what it's all about. And so what we did in here in this sort of in silico system was just do a big simulation study of a range of genetic and management factors across all of these environments. And so this is a big G by M by E. Look at the adaptation landscape. Okay, so we're simulating that adaptation landscape. No way on earth you could do this many experiments to actually look at it in reality. So you look at a number of genetic traits, maturity and tillering, so the amount of branching the plant will affect its leaf area. The duration of the crop will affect how much water it's going to use. Look at those management attributes, that configuration and density, and a whole range of environments, a range of sites, 100 years of historical climate, soils, different sowing dates, and so on. So you've got, a few million crop years, which you can do with this in silico stuff. So it's not really a huge simulation, but it's enough to give you a pretty good idea of what's going on. So if we look at this adaptation landscape, we can use these heat maps to sort of look at a few dimensions. So I'll just explain how this works. So if you're inside a panel here, the y-axis is the tillering. So the top end is high tillering, the bottom end is low tillering. The x-axis is maturity. So this is quick, this is slow. So early maturing, late maturing. The columns here are whether you have all rows planted or you skip one or you skip two. And the rows are different population densities. So your high intensity system is down in the bottom right-hand corner and your low intensity system's up here somewhere. And so if you look at 1 year, you go, okay, there's the adaptation landscape. The peak is over here. Wonderful, I should do late maturity, single skip, low tillering. But if you go to the next year, peak's over there. And so suddenly the whole thing, it's like, that's what I was saying, the thing is a wave, okay? So from 1 year you go, the peak's there, the next year the peak's over there. And when you decide which of these combinations you're going to put into place, you don't know where that peak's going to be. So what can we do? And there's breeding and agronomy involved in this system. And so we've looked at what's the average yield over all of these environments for a range of different genotypes using a standard management. This is what a plant breeder usually does. One management system, look at a whole lot of genetics, and then look at agronomy. How can we adapt that once we've got the standard set of traits? And what we wanted to look at was, well, what happens if you do both at the same time, which doesn't normally happen. So if you run that sort of simulation, you get this trade-off between yield. So these are all the combinations of different sets of traits, tillering and maturity. And each of these simulations is then what's the average yield for a particular combination and how frequently do I go below that break-even line? So that's my risk of failure. And so you don't quite know where a manager is going to sit on here because the trade-off is what's the attitude to risk. And so you will get some increase in production, but you have to accept a higher level of risk. So we've put a risk preference on here and said, well, okay, let's assume that sort of risk preference, some conservative risk preference. Then we can come up with what are the attributes that fit on this frontier, and they are a sort of early medium maturity and a medium high tillering. So we'd say if I was a plant breeding company, that's what I would try to sell you because that works best everywhere. And so if you look at all the regions, we break it down by region and we do the same thing. Here's your yield versus risk for each of these 6 regions. And that's the that's the position that that genotype sits on when you run the standard management. And you say, well, okay, can we modify the management in these regions? And that's what an agronomist does, okay? So in some of these regions, the standard works. In others, you've got to increase the density. In others, you've got to decrease it and go to skip row so that you can get about the same yield for much less risk. And so you can do this sort of, here's my best genotype, let me adapt the management of it, which is what we've been doing for a century. If you look at it all at the same time, then you can adapt the genetics and management simultaneously. Then in some instances, you can do considerably better, and that's the blue dots here. So particularly in these poorer sites, and by changing tillering and managing loss of tillering with density, then you've got a better way to manage the system than you would have had otherwise. And so that's the sort of thing that's an emergent property of this analysis that you wouldn't be able to capture without doing it this way. The point, though, is that that's interesting but useless unless somebody actually does something with it and changes what they're doing. So, you've got to change a decision maker for anything to actually have an effect. And so the interaction with breeders and agronomists and farmers in doing this is critical. And that work is now sort of progressing into application in both breeding and agronomy. So just to broaden this out a little bit, I've probably drilled you into sorghum agronomy and breeding pretty hard. You can use this type of approach for lots of things, and in our group we have done that. So there's opportunities to look at cropping and farming system management along the lines I've shown, but broader than that. You can look at supply chain prediction analysis, how frequently should I plant something and what's the pattern of yield that comes out the end over time. You can do regional scale commodity forecasting, and like the wheat board wanted to know how many boats they should hire. You can look at climate forecasting, you can look at global warming and climate change, and you can look at the implications of those things on what we need to do. You can look at industry policy support, what happens when we have one of those mothers of all droughts, and how do you look at that ahead of time and try to indicate where there might be needs for support. This modelling capability is now a key component of our new ARC Centre of Excellence. That is very much focused on the genetics. And so that Centre of Excellence, which is we're just starting this year, really looks at using this capability as a linking technology between some detailed work on molecular level, on molecular genetics and physiology, on the mechanisms of particular adaptive traits. My role in this centre is in the second part, more about looking at using and developing the models to be able to connect those mechanisms and genomes via predictions into this pathway to crops. And so that's sort of where it's going. I just want to take you back to the beginning and remind you about the hair. So it's sort of all good fun in all those bits, and you can sort of get lost in any of those bits. But it's the integration that really makes the difference, and that requires thinking about it before you take off. And the difficulty with this is probably one individual is not going to be able to do it on their own. You've got all these capacities that you need, and you really need ways of connecting people up and having them understand the language of other disciplines, and not just be able to understand the language of other disciplines, but to actually immerse themselves in it a bit. And so it's not multidisciplinary, it's transdisciplinary, and there's a big difference. So thank you. That's where I want to stop.
Thank you, Graeme. A quick reminder to the audience, if you do have any questions you'd like me to retail through, please post them on the Q&A. tab, and I'm sure there's some questions in the audience as well, which we'd obviously like to have time to hear as well. If I may, Graeme, I've got a couple of questions that have come in at hand, some very detailed, which I'd like to share with you. I'm sure you're going to enjoy answering those, and very specific in terms of the model, and some more general. So first of all, I have a question from Care that's come in on the Q&A tab. And when you talked about the model construct and the general partitioning of components in the model, the question really is around the root system. So the role of the root system architecture. So are there any quantified relationships between the root system architecture traits, such as primary root length, branching density, etc., and yield? In other words, has there been any records of root system architecture as an equation component in the model for making predictions?
Yeah, thanks for the question. Yeah, there's been a fair bit of work on root system architecture. In fact, my group has been involved in that in particular, but so have others. And so, yeah, it's one of the areas of these models that is still relatively naive because of the absence of high quality data. But it was a pity I couldn't show that video of the water extraction, because even with a relatively naive model, which just says roots go down the profile at this speed and extract water on this negative exponential, you can predict water extraction remarkably well. But so you've got to be a little bit careful. Again, there's sort of 3D root system architecture models and all the rest of it out there, and a lot of work on detailed measurements of root systems. The point is, though, there's not a direct connection between that and yield. There's a direct connection between that and how effectively the crop might capture water or nutrient. And that's what you have to be able to predict. And then the model can take care of the rest of it. So I think there is a gap there. I know we've done a lot of work on looking at the angle of the main nodal roots in sorghum, and that makes quite a difference to the root system architecture and its ability to capture water. So there are genetic things that can happen in there, and a lot of people are looking at also not just the physical properties of roots, but their hydraulic properties as well in terms of their hydraulic conductance, which can make a difference. So the answer is, yeah, there's probably something that can happen in there. We probably do okay without it, but you could do better if you knew more.
Thank you, Graeme. Oh, crikey, there's some good technical ones coming in now. So you talked about extinction coefficient, or the leaf area index. That varies with the soil elevation angle, so effectively the solar harvesting capability of the plants. How important, if at all, does the time dependence of the LAI, that is the leaf area index as presented to the sun, how important is that at all, if at all, in the model? I guess heliotropism is probably one extreme of it, where the plants track the sun, but in many cases that doesn't exist. Is that an important factor in the LAI influence?
Yep. Yeah, no, it's a good point. I mean, these models, the way we've constructed them at the moment, run on a daily time step. And so the extinction coefficient is the integral of what the canopy will do over a day. We do have versions of this thing that go back to running inside the day and use the diurnal profile and the angle of the sun and what latitude you're at and what time of day it is, etc. And when we compare both of those, the difference is a few percent of not very much. And so we tend not to get too fussed about it. But there is, you've got to, in making something that is an average extinction coefficient for a day, you need to be aware of when you take the measurements, and that should be an integral over the day. So the measure that you've got for the extinction coefficient takes into account that diurnal variation. Now, if you want to, we're doing work on detailed photosynthesis models, you've got to go to the level that that's suggesting, where you've got to do things at a shorter time step. So it depends, again, what you want to do. If you want to do a broad adaptation analysis, extinction coefficient will probably do it for you. If you want to say, is there any value in changing the structure of rubisco or something, then you probably need to know more about how much of the leaf area is sunlit or shaded at any particular point of the day. And so it's again, it's that, think about what you're trying to do before you run into complicating a model.
Thank you, Graeme. You mentioned soil nutrition midway through your presentation. How detailed is the modelling in Apsim for soil nutrition? And the comment you added is, presumably this is a significant limiting factor: micronutrients, compaction, acidification, non-wetting soils, etc.
Yep.
Did Apsim attempt to take these factors into account somehow?
It takes the main ones into account, certainly not all of them. And so there's been a lot of work on nitrogen modelling in the soil and in the plant, and some work on phosphorus. There's some pH following in the soil model, but certainly, not modelling of micronutrients and the like. So there will be, an ability to model organic carbon and its breakdown over time, and some levels of skill with phosphorus, which was relevant in sort of systems in underdeveloped countries like Africa, where some of your nutrition aspects become fairly critical. But beyond nitrogen and phosphorus, I think probably not. It's an area again where it's a bit like diseases. You sort of, some things that you think, is it really worth trying to model it, or can you just solve it some other way? So I think micronutrients, you've probably, the experimental stuff probably tells you a little bit more than you might be able to get at with a model. And if it's a micronutrient and you need it, put it on.
Thank you, Graeme. A couple of more general questions, if I may, that have come in. You mentioned the ARC Centre of Excellence. Will there be a question on clarification? Will that centre focus on the soil as well as plant function?
It's much more focused on plant function. So it's called the ARC Centre for Plant Success in Nature and Agriculture. So there's an ecology and an agriculture perspective on traits that make plants successful. So it's very much focused on plant traits. Now, some of those are unavoidably going to involve the soil because they're to do with hydraulics and roots. So it's a bit open as to where it goes. But if you look at the chief investigators involved, most of it is sort of plant trait based.
Thank you. Within paddock variation, how can the growers use yield variation within paddock to predict, I guess, yield variability at crop harvesting? What's your experience or your observation of people using Apsim for that purpose?
I think Apsim is probably, I think people have played with it to sort of say, well, if we can zone a field off into a couple of different soil types, what might that do? Or, are there ways of understanding that variation? I think it's again, probably a pretty tough thing to throw a model like this at unless you've got information, the spatial information to be able to drive it. And I know people have looked at it, but it usually just comes down to, well, you can do theoretical studies and say, what if I knew every square metre of the soil had these attributes? What would that mean in terms of advantage if I could manage it optimally for that metre squared across every metre squared in the paddock? And so you can sort of do a what if, what's precision agriculture worth to me? But to be able to actually do that, there's a lot of the stuff in Apsim has been developed from sampling farmers' fields, and so the quadrats of the sample one can predict pretty well. But you've really got all this capacity with satellites and UAVs to get your spatial variation sorted. But if you've got key zonal pieces of soil that you can pull out, then it would probably be able to help with that.
Graeme, towards the end of your presentation, you gave a bit of an indication of, if you like, the users or the use pathways. For example, agri-food value chain actors like, for cropping system management, supply chain prediction analysis, regional scale commodity forecasting. What's the level of sophistication you're observing in the commercial facing users of Apsim?
Yeah, so the level of sophistication is certainly, there's a gradient. So from the plant breeding world, it's quite sophisticated. And some of the commercial entities are certainly well out front in terms of how they use this to help with genetic improvement. As you go to agronomy, I think that gets less so, but there's clear opportunities in that space, and Apsim gets used to look at management by climate forecasting. What are the risks I'm facing, sort of decision scenario stuff. I think when you get to more, we've had some interaction with the sort of supply chain people who might want to say, I want to predict how much tomato I'm going to have every month or something. And normally they don't take the time to collect the data you need to do this properly. So you end up with a less robust system. Now, so there's two ways you can go. You can say, well, okay, if that's the way you want to work, you've got data where this type of technology is not that usable, not that, you can't quite do it. And so you come back to a more statistical approach that says, well, okay, if you can accept, here's the area you've got, and here's the lack of potential to extrapolate to other environments. If you're happy to accept that, fine. But don't blame me when it goes over a cliff.
Yeah, okay. And last question, if I may, noting the time, what are some of the challenges if Apsim were to be used in the context of irrigated cropping?
Irrigated cropping, did you say?
Mm-hmm. Irrigated cropping.
Yeah, no, very good. I mean, certainly, and it's used for that in cotton and other things. So, you can sort of look at scheduling in terms of what's the best way of using available water for a particular piece of land, or at a farm scale, what's the trade-off between irrigating at a certain intensity versus the amount of area you might be able to irrigate given the water you might have. So there are a couple of little software tools that have been developed around those decisions for irrigation work. So I should say that this is the sort of thing that I didn't mention it, but a lot of the stuff I talked about sort of underpins a number of small decision tools that connect into different fields.
Thank you. Look, noting the time, I'm quite happy to bring this to an end, and thank you, Graeme, for your answering the questions. Ladies and gentlemen, thank you for being part of our hybrid seminar. Feel free to reach out to Graeme directly, and of course, with the face-to-face audience in QUT, enjoy the ongoing interactions with Graeme. Don't forget, we'll be releasing a recorded version of this seminar in the coming days, which you'll all be sent an email to advise you where to find it. And just to let you know that our next research seminar, that's mid next month, is going to be looking at the issue of data stewardship and governance. So thanks again, Graeme, for your time, and thank you all, ladies and gentlemen, for joining us.
Pleasure.